Recording biological signals provide a preferential doorway to a wider understanding of physiological phenomena within the human body, either under normal or pathological conditions. An example in this field is the visual evoked potential test. A visual evoked potential is an evoked potential caused by a visual stimulus, such as an alternating checkerboard pattern on a computer screen. Responses are recorded from electrodes that are placed on the back of your head and are observed as a reading on usually an electrocephalogram. These responses originate from the occipital cortex, the area of the brain involved in receiving and interpreting visual signals. A doctor may recommend that you go for this test when you are experiencing changes in your vision that can be due to problems along the pathways of certain nerves. It measures the time that it takes for a visual stimulus to travel from the eye to the occipital cortex. Reading the obtained graph, this measured time refers to latency. Comparing the graphs of the normal and abnormal visions, we observe that the latency is higher in the second case. This indicates that it takes more time for the electrical signals to reach the visual cortex. In fact, this can be caused, for example, by issues affecting the myelin sheath of the optic nerve. Bioluminescence refers to the process of visible light emission in living organisms. In biomedical imaging, it is a technology that uses light emitted by enzyme-catalyzed reactions to report activity at the molecular level. A recent article published in 2021 reports the development of a portable bioluminescent technology and claims that this method overcomes all the major limitations of BLI and provides a major advancement of the powerful BLI modality toward many fundamental and therapeutic applications. In this video, I will only discuss the general concept of the PBL. First, we have the basic principle of functional BLI. A bioluminescent probe with cage luciferin interacts with the targeted biological process. This reaction leads to engaging of luciferin, which in turn is able to react with the luciferase to produce bioluminescent light. Note that the amount of light generated as the result of this engaging is proportional to the level of functional activity of the biological process of interest. In PBL, there are three main components. First, a cage luciferin probe that releases free luciferin. Next, a luciferase biodegradable plug or a cell encapsulating device with luciferase expressing cells that produces light proportionally to the amount of released luciferin. And finally, a sensitive portable light detector for signal quantification. A typical PBL experiment employs an administration of a functional bioluminescent probe, then the injection of a biodegradable luciferase plug. The light production is quantified by the portable light detector that is placed directly on top of the area of the plug. An example that links bioimaging and signaling process is echocardiography. An echocardiogram is a sonogram of the heart. It allows the visualization of the internal structure of the, for example, fetal heart by creating images using the standard 2D, 3D, and Doppler ultrasound. It let us see also the heartbeat signals.